A very warm good morning, good afternoon, or good evening in whichever part of the world you are. I would like to welcome you all to the much awaited event of the year, Omics Research Symposium. Before we get started today, I would like to introduce OmicsLogic. To begin, we must ask, what is bioinformatics? It is the development, implementation, and usage of methods and software tools for understanding biological data. Bioinformatics has practical applications in healthcare, agriculture, and mitigating climate change. As this field progresses, bioinformatics poses several challenges for both students trying to learn it and faculty trying to teach it. In this presentation, I will explain the current challenges and limitations in bioinformatics, describe how we aim to address these challenges with the Omixlogic Portal, describe Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework, explore the Omixlogic Portal and how it utilizes Bloom's taxonomy in structuring its course, pipelines, and projects, and finally describe several outcomes of the Omixlogic Portal. I'm Sonalika Ray, and I'm the OmicsLogic Community Manager. I'm responsible for daily interaction with thousands of users that are a part of our bioinformatics community, which is managed by Pine Biotech. Over the several years, we have had the privilege of working with a number of organizations developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. OmicsLogic research-based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. So what are some of the challenges that the field of bioinformatics is currently facing? To begin, this field is constantly growing and developing new tech and software to improve processing time and power. The data sets that are analyzed are usually very large, and they take immense amounts of computing power and memory that many personal computers do not have. Also, despite being publicly available, data are also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists do not have. In a university setting, students have the opportunity to learn the bioinformatics project of designing and implementing a project, but do not receive practical or hands-on experience with the full workflow due to the limitations that I just mentioned. Similarly, faculty faces challenges with being able to cover a diverse range of topics if they are specialized in a certain area of bioinformatics or if they are unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not know much about the metagenomics of seawater. To overcome these challenges, the Omixlogic Portal was developed. This program aims to standardize, organize, and systemize bioinformatics training in order to make these analyses usable for any biologist or clinician. There are some traditional problems with the standardization of bioinformatics coursework, which includes materials for courses that are usually developed in-house, taught sporadically, and are rarely updated. The Omixlogic Portal introduces new material that keeps up with new technology. These courses need to be organized because the challenge is to combine several different topics together. The flow and transitions between subjects needs to be developed. This is addressed at OmicsLogic through introductory material, which is being woven throughout the courses. The last challenge we face is systematization. How can you know that someone is stuck and needs help or allow someone to run forward? To address this, OmicsLogic uses standardized modules and interactive sessions, beginning with mastering terminology and concepts to implementing these concepts with real data sets in a project-based approach. Here is an overview of the learning process. In order to create a path of skill development, OmicsLogic employs the Bloom's taxonomy levels of learning. This approach starts with the basics, where students recall facts and terminologies and are able to repeat and memorize the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The students will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. So how do OmicsLogic resources help advance bioinformatics teaching? OmicsLogic is a portal for practical and theoretical learning of bioinformatics, composed of courses designed to take you through the theoretical knowledge. OmicsLogic takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give a student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, OmicsLogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or beginning a data analysis project. These 6 courses were taken from the core OmicsLogic curriculum, which is available online, beginning with an introduction to application of bioinformatics and an overview of data types. The student is able to learn terminology and factual knowledge to build upon. From there, genomics, transcriptomics, metagenomics, epigenomics, and data science are introduced. Each course starts with important terms and concepts and then introduces case studies and pipelines related to each one. This allows the students to gain the conceptual knowledge in order to begin their own projects. OmicsLogic expands upon its basic curriculum with specialization track projects. Bioinformatics methods are best learned in the context of real case studies prepared by our network of mentors and collaborators that illustrate how various bioinformatics methods and omics data types are used in current research in medicine, biotechnology, agriculture, and even astrobiology. The projects are prepared by taking publicly available datasets and using them to explore and understand both the research challenge and the analysis path to extract meaningful insights from complex omics data. In this symposium, we will hear from speakers from different domains of life sciences, talking about relevance of omics, integration of omics technologies, advancements in biomedical research, and also we'll hear about machine learning view of multi-omics integration. What we will discuss today and throughout is a result of hard work by an amazing team led by company co-founders Dr. Alfred Tauber, who comes from a medical background, has an extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center at the University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over his course of his career, he has developed multiple novel algorithmic approaches to biomedical data analysis. Under his leadership, the Tauber Research Center started collaborations with research teams at such universities as the University of California, San Francisco, Institute Pasteur, Tulane University, and many others, where research labs are working on cutting-edge research projects studying viral and bacterial infections, and designing novel drugs and vaccines to treat and prevent infectious diseases. The rest of the team includes experts in bioinformatics, computer science, and training. Some of our team members will be presenting today as well. The tool gives students the options for analyzing a given dataset. Code-dependent paths or code-independent paths. These datasets and associated publications can be used in several ways. You can learn bioinformatics without any coding at all. For that, in each project, we explain the analysis pipelines on the user-friendly T-Bioinfo platform for big data analysis. You can also learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in your own analysis. Finally, you can hear about the projects and their research applications in our mentor-guided programs where experts share their experience and support research projects developed by you. Here is an example of the code-independent pipeline. The pipeline builder interface on the T-Bioinfo platform is a color-coded pipeline that helps establish a logical connection between the technical names of data analysis methods and the logical steps needed to build the full pipeline. Data is put in as an input file, and then analysis can be chosen based on the type of research. Each analysis is separated into 3 colors: blue for pre-processing, red for mapping, and green for quantification. Each time you click a button, a pop-up will contain additional information useful for building the pipeline. The ease of the color-coded buttons allow programs to be run without any coding knowledge. Interactive outputs allow easy visualization of the data. Several demo pipelines with real datasets are available for students to practice the workflow, including demos on genomic variant calling of cancer cell lines, 16x amplicon analysis on diet, and its analysis on the microbiome, and a pipeline for examining a phylogenetic analysis of Ebola virus. To answer your curiosity about the T-Bioinfo platform, we have among us researchers for Tauber Bioinformatics Research Center who will highlight few areas of analysis available on the server. Dr. Julia Pano, who will talk about analysis of bulk and single-cell transcriptomics data on the T-Bioinfo platform, and Dr. Elizaveta Taranenko will talk about metagenomics data on the T-Bioinfo platform. After using the T-Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their theoretical knowledge, the remember and understand levels of blooms, and apply that in a project. They apply, analyze, and evaluate levels of blooms. Then check their comprehension with levels and batches, certificates of completion, and test. Similarly, faculty should be able to assign the Omixlogic Portal to their students with an understanding that it will give them holistic understanding and comprehensive knowledge of bioinformatics, beginning with bytes and molecules and onto designing their own question-based project. Omixlogic employs a hybrid approach, combining learning and research to ensure that students are building off of the first level of Bloom's taxonomy. OmicsLogic tracks progress with activities like pipelines, coding, and literature reviews, quizzes on topics, key terms, and important questions, and covers a broad range of topics like biomedical research, biotechnology, and agriculture. Then students are able to continue their knowledge base with a research assessment, making sure they are able to understand methods, challenges, and results, as well as being able to comprehend primary research literature and recreate the methodology. They should also be able to ask questions based on data exploration. This leads to the ability to encounter errors in their analysis, apply theoretical knowledge of why those errors are occurring, and find a solution. Bioinformatics combines life science terms with data science skills, which allows researchers to answer life science questions with reproducible data analysis methods. OmicsLogic is proud to collaborate with many diverse programs across the globe. We also work with over 15,000 students at community colleges like Glendale Community College, top-tier universities like Georgetown University, as well as 20 universities around the world. We are pleased to have among us researchers from our partner universities and institutions who will enlighten us with their talks on different topics relevant to any student or researcher, from knowing about technology and soft skills for industry, skill sets, the missing link between academia and industry, to understanding genotypic data, personalized therapies for cancer patients, and hierarchical patient models. In this presentation, I explained the current challenges and limitations in bioinformatics. I described how we aim to address these challenges with the OmicsLogic platform. We discussed Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework. We also explored the OmicsLogic program and how it utilizes Bloom's taxonomy in structuring its course, pipelines, and projects. And finally, described several outcomes of the OmicsLogic program. Want to know how to get started? The Omixlogic Portal is easy and free for anyone to register with many courses and online meta-guided training programs in the field of omics that includes genomics, transcriptomics, single-cell transcriptomics, metagenomics, and epigenomics. Also, you will find programs in the field of bioinformatics, structural biology, infectious diseases, and precision medicine. These programs are available all around the year, and you can get started with your specialized training anytime at your own convenience. Once you have a specialized training, then you can work on an independent research project, again under mentor guidance on different specialization tracks, which includes the analysis of data types you learned in the training program. These specialization tracks include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data analysis, metabolic disorders, space omics, agriculture, and comprehensive training on omics data analysis. Throughout the year, various students and researchers join Omics Research Fellowship Program and work on their research projects to either gain new skill sets or to complete their college dissertation or for a publication to add to their achievements. Omics Research Symposium will provide a stage to such research fellows to present their innovative research projects. Also, all the research projects will get a feedback from our panel of judges. The OmicsLogic training has been completed by over 24,000 participants from 187 countries in over 500 workshops. And due to this fast growth, our team is now working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. As a result, we have acquired professional certification to offer our curriculum and support research projects using our OmicsLogic project-based curriculum. And here are some amazing stats from an year-long training programs that were conducted by us. The symposium is divided in 3 parts, where we listen to speakers from Asia first, then Africa, and finally the Americas. So to begin with an introduction to OmicsLogic Asia, I would like to call upon stage Dr. Mohit Mazumdar. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals. He has been developing collaborations with universities as a mentor and project lead for OmicsLogic training and research fellowship. Over to you, Dr. Mohit.